Begin the session. A very good afternoon, one and all. On behalf of the principal, the CEO, the management of Patkar-Varde College, I am Ms. Sridevi Devadiga. I'm delighted to welcome you to the RUSA-sponsored international webinar BIOFACET on transforming healthcare through biologics, hosted by the Department of Microbiology and Biotechnology. It is my pleasure to also extend a very special welcome to our distinguished guest speaker, Dr. Surendra Chauhan sir. Encouragement, enthusiasm, excellency are major aspects of our institution, and with similar objective of encouraging the spirit of research and innovation, Patkar-Varde College feels proud to host this international level webinar. Something about a college. Chikitsak Samuhas, Sir Sita Ram and Lady Shantabai Patkar-Varde College of Arts and Science and VP Varde College of Commerce and Economics have been committed to providing a quality education for all since 1964 and has completed glorious 60 years. We are not only dedicated to providing the students with rigorous academics, but we also help them succeed by enabling them to discover their academic passions, synthesize information, and think critically in order to make their career goals meet. Patkar-Varde College is offering graduate and postgraduate degrees, diplomas, and certificates, and PhD programs in various faculties of arts, commerce, and science. Patkar-Varde College has a place of pride among the educational institutes in the western suburbs of Mumbai. In the world that is driven by knowledge and innovation, our diligent and motivated staff has always attempted to improve the quality of education and secured an A+ grade in the NAC third cycle with an institutional score of 3.53 in the year 2017. Following this academic autonomy has been confirmed to the UGC from academic year of 2019-20. As per Education World India College ranking for 2021, our college was ranked 45th in all India and 10th ranked in Maharashtra. The branch of hospitality at Kudal, Sindhudurg district has also become the first satellite education center in Maharashtra that is connected to the University of Mumbai. The IQAC was established in the first NAC cycle of 2003, and since then it has been a vibrant body for our college that helps in integration of various activities of the institute and helps in planning, guiding, and monitoring quality assurance and quality enhancement activities in our college. It channelizes and systemizes the efforts and measures of our institute towards academic excellence. Our college has also been sanctioned the RUSA grant under the component 8, that is enhancing quality and excellence in select autonomous college, and thus joined at least the club of 26 higher education institutes in Maharashtra and has been awarded this grant so far under various components of RUSA 2.0. Di- Introducing our department to you, we are Department of Microbiology and Biotechnology. We organize BioFacet every year, starting from the academic year of 2012 and 13. And it's a stride taken by the department to provide a platform for scholars, teachers, experts, students from various fields of science and technology for academic interactions, elevation, and enrichment. It has also taken initiatives in helping students in extending their minds to diverse fields available in biological sciences. We organize BioFacet every year on a national and international levels and on various topics that include plant health management, biosimilars, vaccine development, CRISPR gene editing, virus research, and several others. We are sure this webinar would certainly complement your learning objectives. On this note, we begin our session of the day with Dr. Surendra Chauhan, an esteemed founder CEO. Let me introduce this person to you. He's an executive board director, brings over 30 years of unparalleled global experience in biotechnology, with expertise spanning drug development, strategic planning, and operational and financial leadership. For more than 22 years, Dr. Chauhan has been at the forefront of advancing cell-based therapies and biotherapeutic molecules for cancer and autoimmune diseases. Throughout his career, Dr. Chauhan has held very important roles, including industry advisor board memberships at very prestigious institutes such as the National Science Foundation's Engineering Research Center for Cell Manufacturing Technologies. Armed with PhD in biochemistry from ATRAC Mumbai and a master's degree from biotechnology from IIT Mumbai, he has spearheaded groundbreaking drug development initiatives at very renowned organizations. Dr. Chauhan's leadership has led to publications of over 50 different scholarly articles and acquisition of several US patents. notably contributing to the development of real-time fluorescent reverse transcriptase assay and cornerstone of modern PCR technology. His research in HIV protease inhibitors has earned him the esteemed Henry Christian Award, underscoring his significant contribution to medical science. Continuing to his excel in global team management, Dr. Chavan remains dedicated to advancing frontiers of immunology and cell biology based therapies, driven innovation and positively impacting patient care worldwide. So it is indeed a pleasure to have you with us today. Before we begin our session, I request all the participants to switch off their cameras and speakers so as to allow a very smooth conductance of the webinar. If there's any technical problem during the webinar, we'll try to resolve them as quick as possible. And kindly post your questions in the chat box, and it will be answered at the end of the session. Now I request sir to please take over the session from me. Thank you so much, sir.
Thank you very much. Thank you, Ruchita. Thank you, the respected professors, teachers of the Patkar College and Varde College, Patkar-Varde College. And I really appreciate, the opportunity you are giving me to speak in front of our young scientist. And I hope and the way I'm going is it's like 45-minute presentation, and we can go slowly. I don't have to complete all the 30 slides, and those slides, Ruchita, you can share with the students also, as well as if they want to contact me anytime, please share my Gmail ID with them. And saying that, what I'm going to do, I'm going to share my screen and just give a one. Just give me 1 minute. Okay. Little bit technical difficulty, guys. Just give me 1 minute. And entire screen. Stop sharings. I think there is some problem with the sharing.
Sir, can we share the PPT?
So one sec, hold on. Let
Yes.
me. Yeah, because I have the what you call. I have some. One sec. Somehow, how do I can do that? Reminder. Enter screen. Okay, I cannot see my screen with the presentation. That's the problem. One sec, hold on. Let me share this way. Hopefully I can get my presentation. No, stop sharing. Yeah, because there are some animations which I wanted to share, or else there's going to be an issue. So, can you see my screen?
No, sir.
Okay. Okay, I hope so. It is there now.
Yes, sir, it's visible now.
It is visible now, right?
Yes.
Okay, and I think I should go into the presentation mode directly. Can you see it properly?
Yes,
Hello,
sir.
everybody. Okay, so the today's topic I'm going to talk about is next generation biopharmaceuticals, and I'm going to just focus
Sir, there
on.
is one request. Can you just change the slide so it is moving? Just we can check.
Okay, is it moving?
No, sir. Yes, now it's clear. Yes, sir.
I
Sir,
see.
you
So
can continue.
I
Yes.
see. So I cannot show the. I think animation is not going to be seen. Looks like we will try one more time. Okay, and see if it is coming up properly there. Can you see anything happening? Like now, I'm going.
No, sir, we cannot see it in
No,
the slideshow mode.
I think you cannot see it in the slideshow mode.
No, no,
Okay,
no.
Yes, sir. We cannot see in the slides no more.
I see. What can be done to
Sir,
really?
may I help you in this?
Sure, please go ahead,
Sir,
Pushkar.
can you stop sharing the screen?
I will. One sec. Hold on. Yeah.
So on the Google Meet button there will be three icons, that is three buttons.
On the top, right?
No, sir, at the below.
Okay.
And over that, once you go to the left hand side, there will be a present now button. It will be a rectangle with a upward arrow.
A rectangle with upward arrow. Rectangle with upward arrow. Okay, present now.
Yes, sir.
Yeah.
Then click on the entire screen.
Okay. I go on the entire screen.
Then select the entire screen again in the
Okay.
box
Okay.
and then OK.
Share. Thank you, Pushkar. Okay. And then go to the screen.
Yes,
Can
sir. We
you?
can see yes, yes, it is now visible.
It is now visible. Okay.
Yes, yes.
Wonderful, Pushkar. Thank you very much.
Welcome, sir.
So, okay. Sorry about this. Always these computers, whatever we say, we are there, but there are always some difficulties. And once you cross the 50, like me, there are some always these technical challenges. So anyway, I will have loved to be present there, so that there will be a good kind of the interaction. So let's talk about this next generation biopharmaceuticals development and challenges. That's what I'm going to talk about. The outline of the presentation is going to be, I will be introducing you about biopharmaceuticals and why we need them. Drug development process, I will briefly give you the overview about that. We will talk about the cell therapies, challenges, solutions, and then we will go through the question and answer. Pushkar, the things are moving properly or there is some issue still?
Yes, sir. It's moving.
Okay, wonderful. So, whenever I present to our young audience, young scientists, I always like to emphasize one thing is that be a truthful and truth will follow you, or else it is going to haunt you. And guys, it's a really very important thing in this current, the generation, or not current, I will say. It is going on for a long time now. The we human race is become like too much self-centered, and as a result, for the some gains we do some, some wrong things. And so always I tell to the young generation, at least be truthful and truth will follow, or else it will haunt. And as a result, there are multiple publications. When we scientists, we are the educated pillar of the society. We publish, and sometimes not intentionally, by mistake, we publish which is not correct. And that's the reason you have to be always be careful whatever you are doing, and you present it, so that when you present something in outside, when you publish, there are like now thousands and thousands of people are going to look at it. And if you represent any mistake, and that is going to be expanded, or it is going to be followed by thousands of people. So that's the reason when I say that be always careful. When I say truthful, that means whatever you are doing, do it and try to understand what you have done. So, to become a good scientist or a good person, what one should be very conscious, very sensitive to the surrounding, what we are doing, to the others also. Be vigilant, curious, and scientific. And always, you young scientists, always, and as well as everybody of us, we have to always ask the question, what is this? Why this is this way? Who has done it? When it is happened? Where it is going on? Or how it is? So we have to ask all this, keep on asking these questions ourselves to get the right kind of the answer. And the answers are there. Only we have to ask those questions. But unfortunately, what is happening is that, we becoming more specialized. And because of that, sometimes specialization, we become blinded with that. So all of us, we put some kind of the blind on our eyes or our senses. I will not say eyes only, the senses. And then everybody know the story about the elephant. It is from the Mahanubhav Pant long back. So how they, blind people are looking at the elephant. But we don't have to be like that. And we have to be always without the blinds. All the senses should be, very active and vigilant. So in short, what I want to say is that be always alert, be always thoughtful, with a keen sense of curiosity and scientific awareness. And that is very important if you want to become a good person and good scientist. And that's how we can build a really good society. And young people, you guys should remember that. So what is happening? See, diseases are always there but we see that there is a increase in the chronic diseases. What are the chronic diseases? When we say that chronic diseases means they are the like continuously you are, suffering with this. And these are the hypertension, high cholesterol, arthritis, ischemic, diseases, heart diseases, diabetes, chronic kidney diseases, heart failure, depression, Alzheimer disease, and all these different chronic diseases, they are really increasing exponentially, and there are almost like 70% people have. Once they cross their age 50 plus to a 60, they will have at least two or more chronic conditions. And what is happening because of that is that the mortality rates are increasing, and they are almost double in many of the countries. So when you look at this graph, there is a change in death. Percentage change in death is on the y-axis and different countries. And the different color bars are basically talking about, what is happening with respect to CVD, diabetes, malignancies, or respiratory diseases. And if you look at that, like in each and every country, over the last 20 years, we see there is a 50%, almost 20 to 50% increase in most of these diseases. In fact, in some of the countries, almost double the percentage. And that is, if you look at the India, and we are also going into the same direction. The CVD is increased more than 50%. The diabetes is almost double in last 20 years. And I feel it is going to be almost we have like 25 to 30% population is a pre-diabetic, and that is really a sad story, which is when we look at it. The malignant neoplasma, and when I say 20%, 30%, that is one in fifth or one in fourth, person you are going to, out of four people, at least one person will have the diabetes. Same thing is happening with the malignancies, different kind of the cancers, respiratory diseases. And that is going to be a major challenge because the healthcare cost is going to rapidly increasing. And when you look at it, and here once again I'm showing on the y-axis the current health expenditure per capita for the different countries and the GDP per capita. So what is the earning capacity of that country basically when you look at it on an average? So when you look at it, there is a red which is on the, I don't know if you can see the cursor there, but United States, which is on the top, where the GDP is almost like $70,000. So they are earning like 70,000, and they are spending around 12 to $15,000 per year on their health. Whereas in India, it is like in 2019, it is around 200 to 200 to $250 per year. Every individual might be spending it, but by 2030, according to me, it is going to go more than 1,000, 2,000. So we are talking about more than lakh rupees per year the people will be, and that kind of the is going to be a big kind of the expenses. So next graph here we are showing the global healthcare spending. So right now in 2020 we are spending around 10 to 15 trillion dollar worldwide we are spending on the healthcare, and that is going to increase to at least 30 to $40 trillion in that range by 2030. And 2030 is coming within a few years, so it is not far away. And you can look at it, the 20-30 trillion dollar, when we talk about it, how big that number is going to be. And this is globally, and in India and US, most of the countries, because of this increased healthcare expenditure, are going to be crumpled. Their economies are going to be crumpled because of that. And we have to think about it, how to handle all these things. And what are the major risk factors? Why we are seeing this increase in the chronic diseases and malignancies, different kind of, you guys are seeing that. And the major culprit is basically the pollution, the way we are treating our air, water, soil, as well as, guys, sound. I know everybody of you young generation are always with those the earbuds and trying to listen to the music, but that is going to be also harm not only on your ear. That there are the hair cells which are called, they are, you are damaging. At the same time, your mental status is going to be in a problem too because of those continuous sound. Then the light is another problem. The too much of the light which we are like at night, there is you don't say anything is dark nowadays, and that also affect on the sleep pattern, and that is going to help affect on the health also. Then the electromagnetic, like we keep the phone next to us, that is become our second organ now. It is one of the organ of the body, and that is another problem which we are going to face. And whenever the major culprits are, like as I said, air, water, and soil, and the contaminating or the pollutant are the pesticides. We are using too many of the pesticides, wrong kind of the fertilizers, industrial pollution. Major cities has got that problem. Industrial pollution, it is really very bad. And then the amount of plastic we are using, and then the small portion is done by microbes, but they are going to be there. But if we keep our environment clean, and then we can control their level very easily. But that's where we are failing. And over and above that, the major another problem we are facing is this fast food habits. The kind of the food we are eating. I'm just showing here Coca-Cola, but that is not. But it's any kind of the soft drink, which is a sugary drink, which we drink. That is another major culprit. It's like when you take 350 ml of any sugary drink, that is giving you like 50 gram of sugar, and you require only 5 to 8 gram of the sugar. So amount of sugar we are, and that is too much sugar is not good. And when you have the too much sugar in your body, that sugar molecules are going to go and hang on to your different proteins, and that proteins are going to then slow down to work because you have put too much of the weight on them, right? And then the other things also, the kind of the cheese which is not our diets. And unfortunately, with the McDonald's, with this TGI, whatever the different kind of the fast food restaurants which are coming in the major cities, and that is going to be one, not going to be, it is already a major problem. So what we should be eating? We should eat really always a fresh, We should eat something with no additives. No additives means we always, whatever is available in the market, nowadays we want to preserve it for a long time. So guys, think always about it. If we put some kind of the additive there or the preservative, and that means that is not allowing any life to grow into that. Now you think about it. If I eat something which is not allowing to grow anything, how does that is going to help you guys? So these are the very important thing when what we are eating, how we are eating. And then another important thing is you should eat always local, local foods. And nowadays we have this habit of eating something which is not, cultivated in our country. We wanted something from the outside, and that is going to be really a problem, because there is a food is a main thing. How we eat, when to eat, what kind of the food to be eat. It's really important, and the timing of the food. I know we are busy, and many of the time we eat at wrong times. For the morning thing, we'll eat at in the afternoon at night, and that is not really thing. So please try and work on your food habits, or that once the time goes, and whatever we do in our young times, that is going to affect after 10 to 15 years. That's how the human body works, guys. So try to pay attention what we are eating, and when we are eating. Then the next important thing is activities, physical activities, and stress. The stress, yes, because you guys are always in the 4 to 6 inches, those screens, that is on the WhatsApp, Insta, and all that. We are unnecessarily increasing the stress, and at the same time, we are not doing any physical activities. In fact, the WHO report, which came up last week, saying that 50% of the Indian young kids don't do any exercise. And that is really, really sad story, you know. And then the another factors are the genes, and then the, our immune system and all that. But everything, these are not in our hands, but the rest of the thing which I just mentioned it to you, that is in our hand. And I hope, in this slide, you try to understand what I'm trying to say and try to improvise your habits. So anyway, coming to the next generation of biopharmaceuticals. So first, we should try and understand the definition of anything when we are trying to, learn. So what is a biopharmaceuticals? These are the complex medical drugs that are made using biotechnology and basically living cells and or organisms. And what is the next generation therapeutics? These are a new way of therapeutic modalities. These are the more complex and diverse than your conventional drugs. And I'm going to give you few examples of this next generation biopharmaceuticals or the next generation therapeutics. Which are those? They are the small molecule like cytokines. These are the very, very tiny molecule in our body. They are roaming, and they are basically communicating different cells with each other. Then the hormones which are there. There are lots of enzyme which require to process and everything. So the people are, when there is enzyme deficiency, we see certain kind of the diseases. So people are making those different enzymes. Then there are the different kind of the antibody treatment for, target a specific thing. So antibodies, monoclonal antibodies, anti-drug antibodies, or the drug conjugated antibodies, fusion proteins, lots of different things which are coming up. Then there are the blood products, platelet-rich plasma, and many other things which people are developing. Then we should not forget about our gene therapies, the amount of research which is going on. And these are the therapies which are going to be like, the way you use aspirin, for example, or the, Dolo, for example. These are the, within next 10 years, you will learn about many of these things, and which will become a common things. Sorry. So, and then the vaccines. So I'm not talking vaccines are there for the ages now. But the new type of the vaccines, like RNA-based vaccines, DNA-based vaccines, the vaccines with the different kind of the additives which are coming, these are the next generation vaccines. And then the important thing is, which is really developing big waves are the different kind of the cell therapies, and we will talk more about these different cell therapies in the remaining few minutes. And then the organogenesis. So one of the thing, as I told you, that because of the pollution what we humans are doing on this earth, many of the organs are going to fail, like the heart, liver, lung, kidney. And literally the kidney, in my village, which is like, we see like 80 to 90% of the people. I am from the Malwan area, and I know in Sindhurga right now there is 80 to 90% of the people will have the kidney disease, in their life. And that is because of the pollution. And think about it, when these diseases become chronic, when there is a problem with the lung, and I have seen many of the people who have this, the respiratory problems. And that is mainly because of pollution. And what else, what we have to do in that kind of the thing. So you require then the organs to be replaced, and which is already people are working on developing the organs. And then the next important thing is wireless implantable medical devices. And some people might think why it is in the biopharmaceuticals, but the way the new next generation these medical devices are coming, they are going to be combination of the devices along with the biologics together. And you might know about the Neuralink now, so that is one of the thing. And then lots of another implants which are coming up into the market. So this is a landscape of the next generation therapeutics which is there. And to go a little bit deep into it, we should know the what kind of the different drugs which we go through it. And everybody is aware of like aspirin, which is a very small molecule, right? It's like 181 Dalton. And when I wanted to explain it to people, I can say it's like a bicycle. Anybody can easily manufacture the bicycle. And the next important molecules comes like insulin or erythropoietin. These are the small, around 5 to 10 kilodalton weight of the molecules, which are little bit complex. They are like a fancy car, basically. Now here you require the more knowledge, more kind of the talented people to build those cars. Then comes your the non-biological molecule, but they are like a heparin kind of the molecule, and they are little more complex than the insulin or anything. It's like you can give the example of the helicopter or something like that. And all these molecule, aspirin or the insulin, LMWH, they fall as a non-targeted therapeutics. When I say non-targeted means once you give it to the patient, they can go anywhere in the body. They are not targeted to one specific location. So they are the non-targeted. And then the next generation therapeutics which comes up, which is like antibodies are there for last 40, 50 years. But now over the time, they become the one of the mainstream kind of the treatment. So there are multiple monoclonal antibodies which are coming up. Then and which are like as complex as like a jumbo jet. So now you require the further more talents, and they are further more complicated. Then there comes your cell therapy, gene therapy, and nanotechnology, which is a highly complex. It's like, it is like universe, which we don't understand anything. We don't understand too much about our simple soil or the water which is next to us. So think about it when we have to think about a cell or gene therapy. It is so complex. It's like you are looking at the universe basically. And in our ancient philosophy, we say that the human being is also has the Brahmanda within in themselves, and which is a true. It is a very complex kind of the thing. And then the next one are the encapsulated cells or the organogenesis. And they are the multiverse kinds of thing. They are the highly, highly complex kind of the thing. And one of the product we are working currently on encapsulated islet cells for the treatment of the type 1 diabetes. So anyway, we will focus mainly on the cell therapy. And before going into that, we should know what is the drug development process, how it looks like. So in drug development, first thing we have to think about is look at the disease. We should understand the disease very carefully. So we have to understand the pathogenesis, we have to understand the mechanism of action, what are the different things whenever the certain thing, for example, the cancer. Is a cancer only because of certain genes, or is it because of certain factors? And what are those factors? And once you understand those factors and the disease, that the mechanism of or the how that disease is generating or inducing, then you have to identify the target to fix those problems which are there, and that's where we identify the target, and that takes around 3 to 5 years, depend on the knowledge of the disease. And these molecules are going to be small molecule or the antibodies or the cells which you generate. You have to then optimize. Once you identify, okay, this is what the molecule I'm going to use, or the I treat the disease, then you have to optimize that lead and then validate really. And when I say this, we will finish it within like less than a minute, but guys, this whole process takes more than 10 to 12 years basically. So when we talk about how to optimize or validate, there are lots of assays one has to develop. Then once you optimize that lead, then you have to you test those molecules in the different animal models basically. So mice, rats, rabbits, monkeys. So that, and one thing, young mind should always understand. See, the thing is that we do all this testing, you know in the different animals, but keep one thing in mind: the animals immune system or animal's body system is completely different than the human being. We are close. We get the information about the toxicity basically from this some of the preclinical studies. And if there is a more interest, and we can talk about some of this thing, and you guys can contact me, and I can help you guys how to look at some of these things. So once the preclinical testing is done, and once you identify, okay, the drug is not toxic and it can be used, then you have to start scaling up. So initially, when you are working, you start producing the, for example, the cells, you are producing it in the small flask, like these are like 20-30 ml. Then you wanted to prepare in liters, and then you wanted to prepare it in multi-liters. So that scale up is very important, and that take anywhere between 2 to 3 years. Once you are ready with the scale up and all that process, you then go and talk to the regulatory agencies, and you file the IND, basically investigational new drug application. You have to file, so that now you can say that, okay, I am some kind of the drug which is going to treat this kind of the disease. I did all the safety and efficacy studies, and I can manufacture in the good laboratory or good manufacturing facility. And once it is done, now the IND give you the regulatory agency give you the permission to take this drug in and test it into the humans. And that's where your phase one clinical trial start. In the phase one clinical trial, you basically look at only 30 to 50 people. You don't give it to the multiple people once, because see, the thing is that when you are studying this molecule, you don't know how it is going to work. You have to figure it out in a few people whether it is a toxic or not toxic. Once you complete the phase one, and when you don't see any toxicity effect, then you go for the phase two. And that's where the multiple, there are now 100 to 200 people will be there in that clinical trial. Then you go into the phase three and phase four, where there are the thousands of people. You test whether this molecule is working, what kind of the efficacy, whether it is really taking care of the disease or not taking care of the disease, and all that. And when you start with like thousands or 10,000 molecule from the beginning, maximum less than a one molecule will clear your phase one, phase two, and phase. And this takes anywhere between 3 to 10 years. And then once you are ready with that, then you go to the FDA or the regulatory agency once again, and then file for the new drug application. In case of the small molecule, in case of the biologics, you apply for the BLA basically. So biological license application, you go for it. And this whole process take anywhere between like, you know, 10 plus years to complete all these things. So what are the ingredients for the success? As I told you, the good observation is very important. You have to understand the pathogenesis of the disease properly. Once you know that, then you identify the lead. And then identify the lead, then you have to optimize and validate it. Preclinical safety studies you have to do, pharma talks and all that. You have to do the production. So you production, then the formulation, which is another important thing, manufacturing, and then the clinical evaluation, regulatory approvals, and then the commercial development which comes into the picture. And to do all these things, lots and lots of time is required. It's between the 8 to 20 years, and lots of money is also required. And in case of the in US, it is like somewhere between 1 to 1.8 billion dollar is required with a current marketing and with current expenses to develop one single drug. So you guys can understand how expensive and how complicated these steps are going to be. And so whenever I say the drug development, to do it, you require tremendous amount of imagination. You have to imagine, and to imagine, you should know the different topics basically. So your basics should be very good in terms of the chemistry, physics, mathematics, and all that. And once you know some of these things, now you have to understand, okay, how the things are happening. It is like what is happening with the cell. Think about it. This is you guys looking at that picture as a very simple picture of DNA and all that. Think about a one cell which is like 10 micron in the 20 micron in size. It has got the chromosome which is like 2 meter in length, right? And now think about that. There are like more than 15 to 20,000 proteins are going to be produced in that tiny space. Okay, and using DNA as the template on based on which the protein is going to be made. Okay, think about it now. As imagine how small the tiny space is. Now imagine, in the morning when you are in rush and you are looking for your, trouser or your shirt in your cupboard. How long it take or the socks, for example. How long it take you guys to identify it? Whereas the cell in that 20 micron space, producing more than 15 to 20,000 protein without making any mistake. And that is happening in fraction of seconds. So think about it, how much things, how precisely it is going on. And those kind of the imagination you guys have to really bring into. And then to have that, then you have to understand, what are the different cell type, what are the different things happening. And not only the knowledge about biology is important, but nowadays, because of the complexity of the different way we are producing the drug, good scientists should know about different science, material science, molecular biology. They should know the applied mathematics, physics, chemistry, electrical and mechanical. See, it is like we have to have the little bit of knowledge about different things. Not only that, we should have the very good knowledge about the computers. The AI, how to utilize the AI is really important. So not only the imagination is important, then the important thing is education, how to execute it. And to do all that, you require really village. You require lots and lots of people. It is not a one single-handedly anybody can do all these things. Then only you can develop the good cell therapy or good therapy. Okay. So cell therapies will quickly go through. I don't know how many minutes I have. I have like another 20-30 minutes, right?
Sir. Yes, sir.
Okay. So, and please, if I'm going too long, please stop me anytime. Okay. So, we will go briefly over the cell therapy, and one should know what is somatic cells and then the somatic cell therapy. I hope so. And the kids, you should understand what is a somatic cells. Okay. So, to give one should always understand the definition properly. So the somatic cell therapy is autologous, allogenic, xenogenic. Now, what is autologous? What is allogenic? What is xenogenic? Autologous is your own cells, basically. Allogenic is from different person cells, and xenogenic is from the different species. When you take these cells that you in vitro or the in test tube, when you propagate them or you expand them and you select them and then you pharmacologically treat it. So you wanted to modify them. And when you modify their biological characteristic ex vivo, means outside the body of that person, and then you administer it back into the human being for the prevention or treatment or cure or diagnosis or mitigation of the disease. And that whole process is called as a somatic cell therapy. So, guys, please always pay attention to the definition, what exactly it is and what does it mean. And then the key milestone in cell therapy, we should know about it, a couple of things that I'm not going to go through each and everything, but the stem cell terminology is developed by Bombori in 1888. And in 1970, the T cells in cellular immunity has been identified. So think about it, in 1970 T cells, in 50 years, now we are developing the T cell therapy for the human beings for the treatment of the cancer. 1981, the embryonic stem cells were identified. 1992, T cells gene editing. All is well? Okay. Are developed. And then in 97 to 2001, something great happened in India, but somehow, nobody paid attention. So, or the one of the first patent is filed by Dr. B.G. Matapurkar in 1997 for the prostate regeneration, you know and in two around the same time, the xenotransplant, that is the pig's heart, is not pigs, it's a monkey heart, is put in the human being by Dr. Barua. But unfortunately, nobody paid the attention to all those kind of the discoveries and finding. But they put the Dr. Barua in the jail for doing those kind. Nobody supported him, nobody helped him. And today, just now, last month or 2 months back, the Genesis, the company in Boston, they did the same thing, and now it has become like a big thing. Yes, we can put the pig's, the kidney into the human being. But unfortunately, nobody paid attention to the people who are doing so much of the great studies in India also. And you guys, the young generation has to pay the attention to all these things. You guys should give the right kind of the respect to the scientist, and that is very important. But unfortunately, we give according to me, these are the four strata of our society. That is, you are the TV stars, or you are the movie stars and the sports stars. I'm not against any of them, but the people who are not as such helping the society, but they are become our icons. But the scientists who are doing so many things, we are not paying attention to them. And then from 2001, the CAR T cell, which has become the main thing right now, and we'll talk a little bit about it. It's become the main thing, and that is started somewhere in 98 around. In 2006, human inducible potent, pluripotent stem cells are identified, developed, and then the first uh, cell therapy for the cancer treatment is developed by Dendron in 2010. And then CRISPR-Cas9, which has become the new thing. In 2017, the first CAR T cell therapy is approved. And now there are like 6 different CAR T cell therapies in the market. And these are the CAR T cells, are the chimeric antigen receptor T cells, which are for the treatment of the cancer. And we'll little bit talk about some of these things. But before going into all these things, we should remember a few things. See, in India, ancient India, we were highly intelligent people, and many of the different might be, but the India, I think, is one of the very developed. And the company name which I like given to my company, which is Rubu. Rubu is one of the ancient rishis who has involved in the cloning of different, animals, and which is properly documented in the Rig Veda, and as well as the Mahabharata. And there are so many other stories which talk about, and people, you guys might be laughing about it, but when you look at some of our temples, the way they have built it, like Kailasha temple in Maharashtra, or when you look at it, uh, that temple, we cannot build it at with the current technologies also. So that kind of the advance our civilization was. So keep that in mind. And then the recently, I come across another article, and you guys might be looking at this tabiz is like, one of the stupidest thing the people used to, put it in their as to their neck, or they used to tie it to their arms basically. And this if you look at that tabiz, the way it is to be designed, it has to have the copper capsule. And do you know what they used to keep inside? Now we completely forgot over last 300, 400 years. But in like 500 years back, the people used to take the the cord, the umbilical cord when the newborn is there, and they used to take it out, they used to dry it, and they used to put it with the ash of the cow dung, and then that used to be put inside those capsules. And whenever the somebody is very ill, those umbilical cord used to be removed and then used to be used as a treatment for that disease. And guys, that is what the modern technology also doing. There are the different companies, they are collecting the umbilical cords, placenta, and they are using as a treatment. See, unfortunately, we had many of the technology, many of the thing. The unfortunate thing is that the many different people rule the India, and we lost our science. So my point is that we should be always proud what our ancient generation or whatever the previous generation has done. We should remember and take it to the next level. Okay, so don't just ignore. See, the tradition in India are very scientific. Everything, when you look at it very carefully, you will understand what science is behind that. But you have to pay the attention to understand that. Okay. Anyway, coming back to the emerging stem cells, and I'm not going to go to all the, there are for each and every different organ, you can have the stem cell therapies, and then the multicellular therapies we are developing. And this is one of the therapy which we are developing, the islet cell therapies, for the treatment of the type 1 diabetes, basically. And they are the more complex kind of the things. We'll not go, but I will give you the little bit glimpse of different kind of the emerging immune cell therapies. And if anybody is interested, we can, have the another talk, or we can, talk ops. You guys can call me and we can discuss about some of these things. So there are the auto allo iPSC derived CAR T, different varieties of the CAR T cells, which I told you, these are the new therapies which are developing for the treatment of the cancer. Then the CAR NK cells, which is the natural killer cells, which are becoming one of the important thing. Then the different kind of the B cells people are developing to take care of the different kind of diseases. The monocyte, which are called as the CAR M cells. CAR stand for the chimeric antigen receptor base, basically. And when you put the T or the NK, that is basically for that cell. And then the dendritic cells. And in India also, there are like 1 to 1.5 million new cases per year are the cancers. And you guys can see that, nowadays the number of cancer cases are really increasing in India. Not only that, we used to not have any autoimmune diseases. But nowadays we see lots of rheumatoid arthritis, multiple sclerosis, IBD, and all these guys, and as well as type 1 diabetes. And all the autoimmune diseases, the main culprit are the pollution, and what we are eating. And you guys really, and IBD, we have seen that there is a tremendous increase in the IBD. We see the allergy. Oh, and we cannot eat and drink the milk after few of the people. And the reason is that the way we are like treating ourselves, and the kind of the fertilizers, the kind of the pesticides, the amount of plastic we are using, all this is leading to all this, many of the chronic diseases, increase in the chronic diseases. We have seen the increase in the neurological diseases, infectious diseases. And another thing is that this cell therapy, in case of the CAR T cell itself, there are more than 1,000 trials are currently going on. And there is a lot of thing need to be done with respect to the cell therapy for the solid tumors, because still the response is very poor in that. So what exactly this autologous T cell therapy? So if you look at it, the first thing is that in case of the autologous, I told you the same person's blood cells are modified and put it back into the patient. That is autologous. In allogenic, you take it from somebody else, you match their HLAs, and then put it back into the patient. So that is the difference between the autologous and the allogenic. So in autologous, you take the patient's blood, then you isolate his specific cells. In case of cancer, suppose you are looking at only T cells, so you isolate those T cells, and then you have to do the modification in their TCR or T cell receptor. And if anybody is interested, we will talk about that in. So you are basically modifying the T cell so that it can go and bind to the specific antigen specifically, and that modification is done by the genetic modification. And to do that, you have to activate the cells and then you do the modification. Once you have this modified T cell, now you have to expand these cells in the large quantity. And that is done by using this different kind of the beads, which are the artificial beads. And then you expanded cells, you remove those beads and then you put it back into the patient basically. And this is what has been done in autologous T cell therapy. And we are the one of the earlier, my company, like almost 20 years back, we started this autologous T cell therapy in US. So what are the complexities of this cell therapy? There are multiple things. It's like specificity is very important. Suppose you are targeting that cell to the specific cancer antigen, then you require that specificity is very important. Another thing is like a potency. So the once you put those cells inside the body to take care of that cancer or anything, now those cells should go and kill that specific, disease moiety. Then there comes into the another challenge which we always face is how to expand, what is viability, because when you are doing all these things in vitro, you are changing these cells so much, so the expansion, viability, exhaustion is another challenge. Memory, the memory is very important. Everything has got memory, cell has got the memory. And unfortunately, when the cells are in our body, they have the different environment, and we take them out and then we grow them onto the plastic ware. So we really change the memory of the cells. So there is a lot many things we have to do so that we can keep the memory of the cells intact. Then the media, because the cells has to grow in a certain kind of the environment, they require certain kind of the nutrient, and that nutrient is very important. So the media is become very important. And once you develop all these cells, you have to storage them, because patient is not going to be ready, or you need the therapy like the Tylenol, you take it. So anytime, whenever you are not feeling well, you should be able to take it. So to do that, you have to store those cells. So the cryopreservation is another important complexity or the challenge. And then the not the least but very important is the toxicity. Is that therapy is going to be toxic to the human being? And then there are the different kind of the toxicities. And we can talk about each and everything like whole one day. And then these are some of the biological challenges in manufacturing and supply chain. There are multiple challenges. One of the important thing is cost and time. As I told you, the cost is too much for these cell therapies, and the time also is required is too much. Then the raw material, talent pool, automation is really the one of the thing which we have to develop for these cell therapies. Quality of this cell therapy need to be very well tested. Storage, as I mentioned, in terms of the biological, there is another distribution logistic. And then another important thing is a IP, intellectual property. And guys, you have to learn about that. Whenever you do any discoveries or identify something new, you should file the IP with the government so that it will be protected. And then the ethic guidelines, regulatory control, enforcement, lot of things which need to be understood or need to be evaluated when we are developing any cell therapy. So there are the different ways you can have the different kind of challenges. I know there is a not enough time, but the potential, as I told you, that you need to have the cell to be doing the right kind of the function. So the potency is to measure the activity of the cells. And what are the activities that the cells are migrating to the target site, whether they can remain in the body for a long time, whether they are going to become lethargic, that is energy, or they are going to be exhausted. So all these things we have to understand before we put those cells inside the body once again. So that potency, so developing all these assays, and you young scientist has to really think about all these things. How do we can improvise all these things? Media, we have to identify the different kind of the media, and many of the thing when I am talking about the cell therapy is going to be the next wave in terms of the treatment, but 90% of the thing we are getting it from the outside. We are not manufacturing it in India currently. And that's where you guys have to really think about that, how do we can think and come up with the different ways, how to develop the new assays to understand the toxicity. See, by doing the toxicity evaluation in the animals is not going to help us. Animals and humans are different. So how do we can taste all these things? What kind of the in vitro assays we should be developing to understand the toxicity properly? At the same time, the storage, like we cannot have the liquid nitrogen everywhere. So we have to come up with the different ways of preserving these cells. And then the safety, how to identify the different kind of the advantageous agent which are present in those final product. So there are lots of challenges and lots of solutions you guys have to develop. The there are the, like, I'm not going to go through, and this is some of the technology which we have developed, the conventionally how you activate the cells. And Ruchita, how much time do I have? Hello, anybody?
Sir, we have 1 more hour.
1 more hour. Okay, no,
Yes,
okay,
yes.
I'm getting tired, but okay, we will talk about few more things, but positively. So
Okay, sir.
yeah. Uh, so the thing is that in case of the conventional T cell activation, what happened? Everything when the cells are getting educated, it happen inside your lymph node. And in lymph node, so there are the antigen presenting cells. So these are the cells which are like a go, they are the not spies you can call, but they are the one who go and check out everything, whether everything is happening properly. When the virus enters, they take the virus or the bacteria, chew it up, and then they present those virus and bacteria onto their surface through this called as a receptors, MHC molecules basically. And there are the police, which are the T cells, which come and check whether this molecule is our own or it is from the foreign, and or it is altered. So that is how the T cells goes and check each and every cell, and this process is continuously happening. And when they find that something is weird, then those T cells get activated. They start producing lots of free radicals, or they try to kill that cell which is infected. And to do that, they produce these free radicals, the cytokines, so that they can call more cells. And that's what happened in vivo when the cells get activated. So, as a scientist, we in like this is in 2000, people decided, okay, how do we can create these antigen presenting cells as an artificial one? So they thought about using this different kind of the beads. And till today, after 25 years, we are still using those Dynel beads, which are called as to activate these T cells. So do we conjugate them with the different antibodies so that they can activate the T cells? But when you are doing all these things, because this is a in vitro condition, see that when the something is happening inside the lymph node, cells will produce all the different required nutrient. Whereas in vitro it is not possible, so you have to provide all this nutrient, and those are the different kind of the cytokine. And one of the important cytokine is the IL-2. So whenever you use this artificial kind of the environment, you have to give lots of IL-2, and then you create these cells basically. So this is in somewhere, when we were developing, and because of some of the challenges, we cannot able to use those Dynel bits, because the Dynel has given their technology to the some other company, for the clinical development. So we were also developing the cell kind of the therapy. I'm talking this somewhere around 2001, 2003. So we decided how to avoid. And I'm telling you this example is that how you should be, young scientists should be thinking about it, different ways of doing the different thing. So we were ready to go into the filing the IND, means basically starting our clinical trial. But the company who owned that technology, they said we cannot use it. And fortunately, we were thinking in parallel line because those dynal beads or those all these artificial beads are too expensive. That time it is costing like $20,000 to $30,000 to when you are producing one of the batch. So we come up with a novel, different technology. How do we can use your own red blood cells? So we started using the red blood cells. We created the technology how to conjugate the different antibodies on the surface of the red blood cells. And we filed like almost 3 patents on that base technology. And this is turned out to be one of the best technology that time when we developed. And you can see that we get the more fold expansion. So the red dots or the red line is basically with the new technology which we have developed, and these conventional bits which people are currently also using, that is in the blue line. So you can see the difference, and the y-axis is basically talking about a fold expansion of those particular cells, whether it is CD40 cells or whether it is a pan-T cells or whether it is a NK cells. So whenever you use this technology, that is by using the red blood cells, because see the advantage of using red blood cells over the dynal bits, there are lots of things, and we don't have too much of time. But when you use a red blood cell, they are like your own kind of the structure they have got. Artificially, when you use something which is completely different, so the cells behave completely different. So this turned out to be one of the major breakthrough when we did that, and that has really helps for us to develop our therapy in better ways. But anyway, coming back to the one of the major challenge in terms of this cell therapy is the complication in terms of the manufacturing these cells. And because of that complication, the cost of the manufacturing is too much. So currently, this cell therapies is costing more than 400,000 US dollars. So these therapies are showing wonderful result, as compared to the conventional your chemotherapy, radiotherapy, antibody therapy. The cell therapy has got potential, but they are very expensive therapies. So, and this cost of, this slide was just give you some idea about cost of this approved cell and gene therapies, what it is going on. So the Hemagenics, this is a gene therapy. The price of that is like 3.5 million. That is almost close to 25 to 27 crore. Okay. Then the next, another gene therapy, which is like almost $3 million. So that is around 24 crore. That is also Zytilozo. That is around 24, 22, 24 crore. 20 crores. Then these cell therapies, which I'm talking about recently, they are also like around $475,000. So that is around 3 to 4 crore. And this is about only the drug we are talking about, not we are talking about the other treatments which are required along with that. So the cost goes to around 4 to 5 crores. Currently in the US and the Europe, the cost of this therapy is in like very expensive. Not the common people can afford it, but these therapies are showing really the very good response, or they're at least controlling many of these cancers. In India, in the Vasi, there is a company, the New Bombay, which is a Immunac. They are developing this therapy, and their cost is also not that cheap. They are also going to charge somewhere around 60 to. They are already the Indian government has given them permission, but the cost is going to be around 60 to 70 lakh rupees. In India also, it is. I'm doubtful how many people can really afford those costlier therapies. So the point is not to scare you guys. The point is that you young scientists can develop the new technologies which can make these therapies more cheaper and affordable to the common people, and that is what is required. And as I told you, the manufacturing and supply chain is one of the major challenge. The cost, financial toxicity, that is the problem with most of all these therapies, is the cost. Like 2.5 to 25 crore, I don't think so, any of us can really start affording them. Manufacturing time is the another major problem. It's taking like 3 to 8 weeks. So the patient is now, there at a last stage. And if you are going to tell, oh, I need 8 weeks for the therapy, that is one of the major challenge. Then having those GMP grade raw material, guys, unfortunately in India, we are not manufacturing many of these things. And you young people have to think about it. How do we can manufacture some of these things? Automation is really important. And because to think about it, like 1.5 million cancer patients we have per year, and so how many cancer patients will be there, and to treat all those, you cannot have the different factories just running by the human being. You require the automation, and then the distribution and all those other challenges which are there. And solutions, as I told you, because the expenses, automation is the key to all these things. And these are the different steps you have to, the cell therapy is a very complex process. And if you can automate each and every step, and there are the multiple companies who are working on, but we have to do it in India. Everybody has to be independent to do all these things. So robotic integration is very important. So one has to understand what is how the robot works, how the computer programming need to do all that. Then how to develop the single use bioreactors, efficient gene editing. That is one of the challenge which everybody is facing right now. Then need the closed loop system, means so that the product will remain in the sterile condition. It will be, so you require the closed loop systems. And then you require the different technologies to monitor how the process is going on. Require the lots of different assays. And there are the another important challenges are the intellectual property. These are very complicated things because of the way the science is there. So it is a very complex. So we require the lots of good lawyers or the people who understand the IPs. So there is a lots of scope to you guys to look at how to get into this patent law. And because of this, all the cell therapy is a complex process. It is not a single thing. And there are the regulatory challenges. So we require good regulatory people. So you young kids has to really think about it, how you guys can do that. There are lots of ethical challenges going to be. Whenever you develop something like this, ethical challenges are going to be there. And then the healthcare cost, as I told you, that the cost, how to control this cost of the therapy is really important. And the main thing is that, as trying to tell you guys, the talent pool is really important. Young minds has to really work very hard. And to do that, we require lots of cell therapy scientists, lots of physicians, lots of laboratory personals, omic experts, bioengineers. We require immunocell biologists. We require the bioinformatics guys. We require the CMC. It is to understand this whole process and then develop the different kind of the critical, testing analysis. So we require those analysts. We require the QA/QC person. We require the microfluidic people who understand that. We require the AI expert. And then we require the healthcare worker, regulatory people, insurance policies. So there is a lots and lots of talent is required, lots of things to be done. So one of the things we have to do, take home message is that, first of all, I know you might understand the story that we don't have to be a blinded people. We have to be always go above and beyond. We have to be there, always be, open with eyes, open with all the senses to look at what is going on around us. And with a practice, try to be Trikalnani. When I say Trikalnani, that is one of the Sanskrit word. We should know the past, we should know the present, we should know the future. And the reason is we scientists really build the societies, we build, nations. And scientists, see, the problem is the plastic. The day somebody has developed that plastic, we thought this is one of the greatest, discovery for the humankind. And today we are suffering. The problem is that we never given the complete thought for that. We developed the great pesticides, fertilizers, but the problem is that we never thought what is going to happen. Right now the guy is the DTT, one of the pesticide which is developed in 1940s, and today, we human beings are suffering because of that DTD. That is what is one of the major concern for the type 2 diabetes. And that is, you see in second or third generation. So when I say the Trikal Jnani scientists are really important, the scientists who thinks about a future, long-term future, not the short-term future, and that's what is required. And to do all these things, my really sincere request to all of you, young scientists, to read the different books. And there are a list of the books which each and every biotechnology student should read it at least once, like Molecular Biology of the Cells, a Biotechnology Concept Application by Clark, Biotechnology for Beginners by the Renberg, Leninger. I hope you guys are reading these books. Genes by Benjamin Lewin, Introduction to Bioinformatics. Then the AI is really going to be, and this the Y Fatak and Saikia, they have written this book. It's one of the wonderful. Try and get and understand what is going on. And another important book I always recommend to everybody is that read the Rigor Mortis. It's one of the wonderful book. How the sloppy science create a worthless cures, crushes the hopes, and waste billions of dollars. So my point is that be always vigilant, be the curious. That's what I will say. And please feel free to contact me anytime. Ruchita ji, please share my Gmail ID also with the people. And I'm ready for the questions.
Yes, sir. Thank you, sir.
Participants can put on their questions in the chat box, or if it is possible, you can speak up.
I think nobody is going to ask the question. I have to ask the question. Somebody to explain me then. What is the difference between the somatic and the gametes? Wonderful.
I think we have one question. How we can decide what topic to choose if we plan to do PhD in bioinformatics?
Very nice question, Abhishek. In bioinformatics, there are a lot of things, guys. Lot means a lot of things going on. But you can think about it, gene. The way the genes or the way the whole the molecular biology, like proteomics, the way it is going on, the way the genetic changes which are happening. So if I understand you correctly, what topic to take it, there are a lot of things in terms of the field is exploding currently in terms of the gene changes, whatever is happening, or the amount of the research which is going on. It's amazing. You guys can look at one of the platform called as the NextNate, the company who is developing the tools to develop the good drugs by using the bioinformatic platform, basically. So, and the name of the company once again is the NextNet, Abhishek. And to answer you, you can go into the genetic, or you can go into the proteomic, or the glycan biology also is becoming a very important. So there are lot many things you can do, you know. So you have to develop your own interests so that you can decide what to do. Okay. Is there any other question? I know I saw some of the questions regarding the type 1 diabetes and some of the other things. Okay, that's a really good question. Somebody is asking the question about how authentic it to use the AI. See, it is always I say that, and that's the reason I said the truth, be truthful, and truth will follow you, or else it's going to be haunting you. The reason is that many of the publications which are there, because our AI is based on what we humans have done in the past, and that is we are putting in terms of the database, and then it is going to give it us back. Okay, so no doubt AI is going to end and that's where the next net. If you look at that company, you will understand what those people are trying to do, and I'm trying to help those guys to develop the good platform. But that everything is, guys, going to depend on this AI is what we have done in the past, and that is what we are going to get in the future. And that's the reason you guys, whenever you present something or publish something, you guys have to be very critical about what you are presenting, how you are publishing it. Okay, if you are not clear, see, the negative results are also the results, and that's unfortunately the things happen in the past. Sometimes the many of the people, if they do the 10 experiment, one experiment work, they only publish the one experiment which worked. And that's the reason we created lots and lots of garbage in the database. So hope it is going to be evaluated and changed. But my suggestion is that whenever the AI gives you some answer, don't follow it. Use your logic. Try to do a critical experiment, critical evaluation. Talk to the right kind of the expert, and then only go with it. Okay. Anything else?
So there is one more question, like what kind of software or websites can be used for the drug development?
Oh, there are not yet good, website. That is one of the. It's a really good idea, Abhishek, and one has to develop those kind of the website. But there are right now, right now to my mind come is the NextNet is one of the company who is doing really good job. But the thing is that still they are not developing that. Okay, what are the different steps? You have to first understand the drug development process, and there are the small modules are available, Abhishek. One can utilize those modules. Suppose and that you want to know, okay, certain genes how they are expressing in different cells type. So there is then the NIH platform is there. So you can go there and try and identify, okay, what are the different genes where they are expressing. Then you want to know the toxicity, whether some of the drug, what kind of the toxicity is known. And then you can go to those some of the website, and there are the enzyme pathway related website. And if you are interested, I think I have the some of the list of the website. I don't mind sharing it to you, so send me the email. And there are the ways you can go to this, but right now there is not a single platform. And that is one of the idea this next net is we are trying to do it, is that how to bring all these different platform, under the one single umbrella, so that you don't have to go to the 10 different website to look at it. How do I develop the good drug? And one important thing is you have to really understand the process of drug development. Lots of things, we cannot cover in 1 year, 1 hour, what has been done by, millions of scientists over the last, 100 years. Okay.
So there's one more question from Senjuti Biswas. What can we use instead of Schrodinger software?
What can be used instead of, sorry guys, I don't know about it. What software you are talking about?
Schrodinger software.
Sorry, Raj, I'm not, I don't know about it. I don't know, but
Another,
I
there's
cannot answer
another
you.
question.
Okay.
Will cancer ever be cured by the help of a vaccine or other forms of medicines? or are only ways to its cure?
Okay, it's a really, really very, very, very good question. Okay, and I'm going to take a couple of minutes. Okay, see, the cancer is your own cell is going haywire, right? See, the thing is that this is a law of our nature, that expansion. Everybody understand that, like law of entropy, enthalpy, and all that. Uh, so that is a basic concept that everything wanted to grow, everything wanted to, disperse basically, and that is what it is in our cells also. So the day we are forming, in the embryo, when we start forming. See, the thing is that every cell wanted to go crazy, no doubt about it. Okay, but there is a nature we want everything to be under control, so that we develop this immune system. So every cell wanted to become grow like crazily, but the immune system and many other factors keep them into the control. So when we start seeing the symptoms, or when we see that there is something, kind of the cancer is developed, so our immune system is basically failed, or the cancer is become over smarted our whole defense mechanism. Okay, now it is your own cell. So first of all, when we use the word the cure for many of the things, like the viruses who integrate into the cells, it's a wrong kind of the word to use, that I can cure certain those infection, I can cure the cancer. We can control the cancer. See, you can have the better life, you know you will not be suffering with that cancer. And at the same time, so please don't guys use the word like can we cure it. It's the cure is not the word, but you can control it, you can live with it without too much of the problem anyway, and that is going to be possible. Now when it is a really good thing, people are thinking about, oh, can we develop the vaccine? See, I'm of a different kind of the opinion. See, the cancer is not because of the single kind of the change which has happened. So when we talk about a vaccine, we talk about 1, 2, 3, 4, 5, maximum 10, 15 or 1,000 different antigens we can target. But a cancer is not. Cancer is because of multiple things which are happening. So vaccine, yes, for the common thing, we can have the certain vaccines, but for the cancer of different things, can we control it? It's going to be a challenging kind of the thing. Okay. And if one of you guys make me wrong, that will be really, wonderful. But think what cancer it is. That's what I tell everybody whenever we come up with some kind of these absurd terms like, yes, can we cure? It's not. We can control it, guys, positively, because it has become your part, or you have to uh, change that organ completely, or change that, and then it is going to be possible if you can identify each and every cancerous cells, and which is possible one day. But the vaccine and some of the things, I think it is challenge, going to be big. I hope you guys understand what I'm trying to say is that, so cancer is not that simple disease. That's the reason so many, we cure cancer in animals without any problem, and that to a certain extent. But because it is your own cell which is going crazy, and how to identify and then kill all the different cells hiding in different locations, and when we can achieve that kind of the thing, and which is possible, I'm not saying it is not impossible, which can happen in 50 or 100 years, the way the technology is going on, certainly.
So
Okay.
we have few more questions. What are the safety and long-term results or considerations for biologic therapies?
Okay, so see, the thing is, it's a really good question. Once again, you guys are asking, a very good question. See, the thing is that as long as we stay with the proteins, okay, as long as we are not doing too many genetic changes, and it is not becoming the part of our gamete, you know, so we will be okay. As if we transfer it to our next generations, in the following generation, what is going to happen, we really don't know. And those are my always concerns about some of the gene therapies. As long as we stay with the somatic cells, We will be fine with it. But the thing is that there are the certain challenges which we have to really understand. That's where you guys have to really think from the futuristic perspective, and that is my one of the, I know it is a weird idea, but I always feel that in ancient time, if those people were so smart and they have developed, I think some of those development might have wiped out our human race also some time back, which is really good possibility. And that's the reason we have to be very careful when we develop some kind of the gene therapy, what kind of the side effects are going to be there, and that's where you guys, you young generation, has to really think about it. Don't think only for the short term or within like a one generation of what kind of the effect of it is going to be. Try to evaluate or come up with the technology which can help us to understand in 3 4 5 generation what is going to be happening, you know so to answer your question, as long as we stay with like protein related therapies, I think we are okay, as long as there is not we evaluate the immunotoxicity very well, and that is another whole chapter. And guys, it is difficult to answer you in quick succession. If you guys have really interest, we will talk about immunogenicity and toxicity in subsequent lectures, if you guys allow me to talk sometime. Okay.
So we have a few more questions. What are the applications of omics in medicine, and what specialization on a master's level can pave way for us to enter the field involving therapies and so on?
Oh, the omics is really the wonderful thing. There are a lot many things, guys, and now we understand how the certain mutations, certain changes can influence. So, at the master level, also, guys, there are lots of opportunities, job opportunities, because the way the bioinformatics is also growing, that will really help you guys. And the omics really helps nowadays, because we are developing these personalized cell therapies, right? Personalized any therapy, not cell therapy only, personalized medicine, which is developing. And that's where it will really be helpful. So there is a great job opportunities for the omics, no doubt about it. So think about how to integrate many of these things properly and learn it.
So there's one more. In future, sorry, what specialization? So how is biologics being used to combat antibiotic resistance?
Okay, that is really a good question. So we filed 2 years back one of the patent about, the resistance is one of the thing, guys. Drug resistance in case of the bacteria, in case of the any sector. The resistance is where the evolution is continuously happening. So the bacteria, viruses, or any of those things, when they expose, there is a certain modification or mutations which are happening, and then the survival survives, right? Now the question which you are asking me, how do one can use the biologics? See, the one of the patent which we filed, I think 2 years back, is that you can target multiple different site by using the, for example, an antibody or aptamer, the different location of the virus or the bacteria. Now when you do those kind of the ways to target any virus or bacteria or fungal or this pathogen, now that pathogen is not going to do the changes, multiple changes simultaneously. So that is how one can really use biologics, or the biological drug development strategies to overcome some of these drug resistance issues. I hope and you understand what I'm saying. So you have to target multiple site on the organism. It's a good question, but
So one last question, sir. How will AI helpful? How will be AI's helpfulness in reducing cost of such expensive tests? Because we have to construct a platform specifically to help in the medical field from scratch. Won't that lead to a further increase in the costing?
That it's a really good. You guys are really asking very good questions. The cost, we can bring it down, guys. It's not that problem. The problem is the society, the way we have built a society. If you look at the major company CEO, and they are earning like a humongous amount of money. It is not the cost of manufacturing. See, the thing is that technology is there. Look at for example, insulin. The cost of the insulin and the technology has developed so nicely. The cost, whatever was there, 10 years back, in fact, the cost of manufacturing the insulin is gone down by almost tenfold to 15-fold of manufacturing. But the price of the insulin is gone up by 20 to 30%. See, it is in the society. Whoever has asked me this question about the cost, how the AI. See, it is not the AI, it is our mindset we have to change. And that only the way we can change the cost, And the cost of many of this drug, I know it is like 3 million, like 25 crore to like 2, 3 crore. And that is also going to change over the time, guys, but it is not going to be the effort by the scientists. It is going to be the effort of the whole society which is needed for that. And that's where you guys have to really think, if whenever we are talking about, the cost, why the cost of certain thing is that much. It is our greediness of the human beings. It's nothing to do with, the AI or anything like that. And that the reason, everybody, we have to change our mindset. Okay.
So one last question. What is the future of nanomaterial based biosensors for diagnosis of early cancer?
It's a very good, the see that the nanotechnology and all these things are going to be, it's a really good thing. But once again, whenever I say that this new technology are really important and they have the great potential, but always think about from the toxicity perspective. Whenever we are developing some of these things, what are the long-term effects? But yes, nanotechnology, and guys, this nanotechnology, nanomer, many of these things are there for the long time. Only nowadays we use those terminologies, nanotechnology and all those kind of the thing. Okay. But yes, there is a tremendous potential, but only think from the toxicity perspective and long-term effect once something is there. Okay.
There are no more questions. Let's proceed towards the valedictory function. I, Mr. Abhishek Das, on behalf of Patkar-Varde College and Department of Microbiology and Biotechnology, feels heartful of gratitude and admiration for the remarkable journey we have undertaken together in this enlightening webinar. It is truly an honor to address such an esteemed gathering of individuals who are deeply committed to advancing our understanding and working tirelessly to find effective solutions. I would also like to express my profound appreciation to our distinguished speaker, Dr. Surendra Chavan, who generously shared their experience, insights, and breakthrough findings. I begin my vote of thanks by addressing our appreciation to the management of Chikitsak Samuha's Chief Education Officer, Dr. Mala Kharkar, our Principal, Dr. Pratibha Gaikwad, Chief Coordinator of Sciences and Coordinator of Microbiology, Biotechnology, and Bioinformatics, Mrs. Ruchita Dalvi, and to our sponsoring organization, RUSA. I extend my heartfelt thanks to each and every participant who joined us from around the world, bringing with them a wealth of knowledge, diverse perspectives, and unwavering dedication. Your presence here today underscores a collective determination to conquer this challenge. Behind every successful webinar is a team of dedicated individuals who work diligently behind the scenes. My gratitude goes out to the teachers who ensured the seamless execution of this event, allowing us to come together in a virtual space where ideas and knowledge flow freely. As we depart from this webinar, let us carry forth the torch of collaboration, innovation, and empathy that has illuminated our discussions. The participants will receive their participation certificates through mail once they fill the feedback form, the link for which has been posted in the chat box. Please note the link will only be active for 30 minutes. In closing, I want to thank each one of you once again for your unwavering dedication. Thank you.
Thank you very much. Thank you. And please feel free to contact me anytime, guys. And send me the email. Okay.
Thank you, sir.